Hi everybody, welcome once more to Salimz Learning Simplified. And if you are the first time visitor of my channel, keep in mind this channel is meant for helping and simplifying various things that the students should learn. So guys, if you are an enthusiast to learn by using tricks and tips, you are at the right place, and go ahead. If you have never subscribed to my channel, just subscribe right now so that you can get specific subjects and learning methods and methodologies in a very simplified way. So guys, today what is our topic? We are going to discuss about dihybrid cross. So without any delay, let us get into the topic. Come on, let's go. What is basically a dihybrid cross? Let us check out exactly what's it. Look, Gregor Johann Mendel has done so many crosses, as you know. The first one is monohybrid cross, the second is dihybrid cross. When he studied a cross in which he took care of only one particular feature or character or trait, we could call it as monohybrid cross. When he took two characters together in a cross and studied, we would call it dihybrid cross. So in dihybrid cross, it is essential that we're going to study two characters together. And look guys, the board is not enough to completely do the cross in a single shot. So I'm going to divide the board into two halves, just like imagine two pages of your book. So accordingly you follow. So let me divide the board into two areas. Okay, and here this is the first page, second page. Now let's go ahead with the crossing. You know, there is a pattern of crossing in which we have to always write the following stuff: phenotype, then we go for genotype, then gametes, and then we go for the first generation, and there finishes the first generation. And then again we have to go for next step, that's called selfing. So we gotta give the heading selfing here, and again we go for writing these steps once more. They are phenotype, genotype, and then gametes. And once we finish gametes, just keep in mind it's so difficult to write all the things over here, so I will just get into the next page. And remember, this is the margin which I have drawn right now, and we go for F2, which is always done in a Punnett square, okay, or also called checkerboard, designed by the scientist called Reginald Punnett. And then after doing this, we would go for a special section called phenotypic ratio. All right, so with that we are going to conclude the topic. Everything comes under this particular headings. Look, what is the beauty of arranging like this? You are not going to miss any particular topic. So this is a standardized pattern of writing any cross. So we have to write phenotype, genotype, gametes, F1, phenotype, genotype, gametes, F2, and the phenotypic ratio of the F2. All right, let us get into the details right now. So of course the cross is done between a male and a female, so we have to show that there is a male and a female. These are the symbols used in biology to show a male and a female. The two major features Mendel took to study here, the two major traits Mendel took to study here are the seed shape and seed color. Recollect, seed shape and color. And then what is shape? There are two shapes. What are they? One is round, which is dominant, and one is wrinkled with some wrinkles on it, which is recessive. And then seed color, what are they? One is yellow, which is dominant, and one is green, which is recessive. So the two features studied are seed shape and color, and the contrasting features are round, wrinkled for seed shape, and yellow and green for seed color. Remember, round and yellow are dominant, wrinkled and green are recessive. So now it is our time to understand exactly how to do the cross. Guys, do not skip the video. Check carefully, slowly, step by step, so that you gotta catch exact method of writing the dihybrid cross. So let us dive in. Okay, number one, the phenotype. Phenotype is the external expression of the character of that particular plant. So the external expression, or how the plant looks like. Okay, so regarding the seeds, we have round as well as wrinkled. So one seed taken, for example, in this example, the male has a round, whereas female has wrinkled. Sorry, round, and female has wrinkled. And then there are two characters required. Only one character we wrote. What is it? The seed shape. And now we have seed color, yellow and green. Okay, let us go for yellow, both the dominant features for male, and green for female. So our phenotypes of the parents used are round yellow for male and wrinkled green as female. Let's write the genotype. As you know, the factors written by Mendel as alleles or pairs are always called genotype. And let's go ahead. Round yellow. Round should stand for a pair of factors. As you know, every sexually reproduced organism is made from a pair of parents. So certainly they're going to have a pair of factors, isn't it? Okay, so here we go. Round is capital R, a pair of factors, and yellow we can give capital Y, capital Y. We use capital letters here because round as well as yellow both are dominant. If they are recessive, we are not going to use capital letter. So here we are seeing wrinkled and green. Are they dominant? No, they are recessives. So we are going to check them and give them small letters, small r, and small y. So this is particularly called as genotype. Come on guys, keep in mind, if we are learning only one particular character, we may have only two letters, and that's called monohybrid cross. But here we are learning two characters, that's why we are having four factors. All right, and this is completely dominant, this is completely recessive. The technical terms used here for completely dominant is homozygous round yellow, and this is homozygous wrinkled green. As you know, homozygous means all the alleles are same. Now let us go for the step 2. What is it? In step 1, generally people don't make any mistakes. Okay, so come on guys, listen, follow carefully. Step 2. What is it? Here we are going to write the gametes. In this particular step, when we write the gametes, some people, at least 5% of people, make mistake, and hereafter everything goes wrong for them. So just keep your eyes open. So the gametes must take only half of the chromosomes. You know that the human beings have got 46 chromosomes. If father is planning to give all the 46 chromosome and nothing from mother, that's impossible. Mother also equally have to give the chromosomes. So as you all know, 46 and 46 from two, parents, mother and father, gives 92 chromosome. It's not going to be a human baby at all. You got the point. So due to meiosis in father's testicles and mother's ovary, these cells undergo reduction, division, and make the sperm, which has 23 chromosomes. Here also the egg, which has 23 chromosomes. That means haploid condition, as you know it. So here, out of these two letters, only one is allowed, because half only allowed for the next generation. So here we go, one letter from R, and we can take one from Y. That is the gamete for the male. And here we go, one letter from both the letter types, and that is a gamete for female. Remember, to identify gametes easily, to distinguish from the other genotypes especially, better is to put a circle around it. Okay, that is next called F1 generation, and we can just cross them. Remember, while writing the result here, 5% of people make mistake. They write capital R, capital Y, small y. No, we are supposed to write R first, the round pair and the yellow pair. Okay, so don't write capital R, capital Y, small y. Rather, write complete alleles of round, then yellow. And that is, of course, if there is one capital letter R, it says it is round. There is one capital Y, it says it's yellow. So this plant is, of course, round yellow. And we are supposed to write the genotype in bracket suddenly. Okay, some people forget to write this, and sometimes people lose marks, half mark or something like that just because of forgetting this particular step. So guys, this is only the first cross and the production of F1 generation. Now let us go for the second part. As you know, F1 should undergo selfing in order to get the second part, and that is the part 2, where the F1 offspring is going to act as the father as well as mother, because it is called selfing, self-pollination. While in this particular point, remember, when the question is about animals, the crosses, okay, do not get yourself into selfing, as animals are either male or female. Same organism cannot reproduce by himself, okay, except hermaphrodites. So guys, let's go ahead. Now selfing is between this same F1 as father as well as mother. So under the father column, we can write round yellow, okay, father is going to be round yellow, and the mother, okay, this X mark says cross, okay, mother is also round yellow because that's called selfing. Now what is the genotypes? We have already written here, okay, capital R, capital Y, small y. Same way here also, capital R, capital Y, small y. Now guys, gametes. Here when the students write gametes in this F2 generation, coming to the F2 cross, most of the students make mistake here. Okay, so let's understand and look carefully. I'll just tell you how to write gametes here, and later on I'll rub it out. Okay, so it's nothing to work out in this page, but just to get an idea. Look over here, capital R, capital Y, small y. They are going to make gametes. How? The capital R could take a chance to go with capital Y. Am I right? Let's go for capital R's equal chance to go with whom? Small y. And then let's try another one. Small r could go with capital Y, and small r could go with small y. Here, by taking this way, come on, let's try capital R with capital Y. Okay, then capital R goes with small y. All right, and then small r going with capital Y, and smaller going with small y. These are the 4 gametes for one of these parents, and then we have yet another parent. So let's go ahead for their gametes, same like this, capital R, capital Y, capital R, small r, capital Y, sorry, we have to write capital Y and small r, small y, and that's all. And this is the major step where people make mistake. So use this method and write your gametes. Never tumble up during the exam. Now let's go for writing. Look, in monohybrid cross, normally teachers draw diagram lines like this and connecting it, but here we have to draw and find in a 4 cross 4, 16 chances are there. Drawing all these lines will mess up things over here. So guys, we will just go into a fresh page and work out how to draw and find the F2 generation. So for that, what shall we do? The specific method designed by Reginald E. Punnett is called Punnett square. In that, we're going to draw a particular table. Okay, and here we have 4 gametes, 4 into 4, 16 are the results. So I'm going to make 4 rows, 4 columns. All right, so now we made 4 columns, and let's go for 4 rows. Okay guys, so we have 4 rows and 4 columns. Look, after that, put a line like this. So this says is for male, this is for female. Okay, let's write the symbol for male here and female here. For those who confuse, this is male and this is female. All right, now once we have written this column, checkerboard is ready or Punnett square is ready. We're going to put all these 4 gametes over there. Okay, so let me write them: capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. That is choice 1. Guys, while writing these letters, you should make it very clear that capital and small letters are distinguished perfectly, or else at the end, the high uppercase and lowercase, you'll confuse like hell, and while writing you are going to mess up things. So be careful. So let us write all these gametes over here also under the females, and do not forget to circle them. Okay, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So guys, that is how we bring all the gametes and write on the sides. Now let us go for all the possibilities from here. Let's now, while writing here, so many people make mistake. Remember what we used here. Same way, R pair first, then Y pair. Okay, remember R pair first, then Y pair. So R and R, capital R, and capital Y. Okay, never write mixing them up. So that's first. Let's go for this one. Capital R, capital Y, small y. Yes, along with me. Now before I go, I'm gonna fill this box. You can tell with me. Come on guys, try. You're gonna do that. Capital R with small r and capital Y with capital Y. You're correct. Let's go for the next one. Capital R with small r and capital Y with small y. Always give importance for capital letter in one pair. So let's go capital R, capital Y, small y. So guys, maybe you're working faster than me. Let's go. Capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y. You notice here small y, but I'm taking capital Y and writing first. You know, you got the point. Always write capital letter the first. And let's go for the next one. Capital R, small y, small y. Here we go for the next. Capital R, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y. And last row. So capital R, capital Y, capital R, small y, and we go for small r, capital Y, small y, and last small r, small y. Now we have written all the possible generation, that's all the possible types of genotypes going to happen in F2 generations. F2 means filial 2. Now guys, we are going to find out what are the phenotypes of this particular genotype. So let us check. If at all you read a capital R, means it is round for sure. To become wrinkled, you need to see both letters as small r. Hope you understand what I mean. And to find yellow, one capital Y will do. But to find green, you need to have both the letters Y, small case. Okay, so here we go. Capital R means round, capital Y means yellow. So I got to write the first of my product, round, yellow, and let me write number 1, tally mark it, and don't forget to tick it. Okay, let's go. Round and capital Y means yellow. Okay, I'm going to put second tally mark, round and yellow. Okay, third tally mark. Now you can work along with me and come on, tell. Capital R means yes, round, capital Y means yellow. Okay, we got to take 4. Now capital R means round, capital Y means yellow. We got to take 5. Capital R means round, but no capital Y. So what is this? Small y means not yellow but green. So we have to write the second one type called round, green, and I could put a tally mark there. Forgot to put here. So let's continue. This is capital R and capital Y, both are there, so it's round, yellow. Now capital R but no capital Y, so it is round, green. Very good. So capital R with capital Y means round, yellow. Now capital R with capital Y, again round, yellow. Here we can see no capital R, so that means it's not round. It has to be wrinkled, and capital Y says yellow. Wrinkled, yellow. Let me put 1 and let's go for the next. Okay, I have been forgetting all these things. Okay, next again, small r, capital Y, wrinkled, yellow. 1 more. Let's go for this. This is round, yellow. Capital R, capital Y means round, yellow. Then round, green, because yellow is missing. Now wrinkled, yellow. All right, and this one, look, both are recessive. That means it's wrinkled as well as green. So I'm going to write the next category, wrinkled, green, 1. And now we have our ratio. Let us write together 9, 3, 3, 1. So we get a phenotypic ratio of 9:3:3:1. So guys, hope you understood exactly what is dihybrid cross and what are the major steps where students always make mistake. And some analysis on this particular table gives you some beautiful stuffs. Come on, look all the way like this. In this particular diagonal, you're going to see the letters are same, but all are for round yellow, round green, wrinkled yellow, wrinkled green. Wow, let's go all the way this diagonal. Hey, oh my god, all are heterozygous round yellow, capital R small r, capital Y small y, capital R small r, capital small y, isn't it? So they all are heterozygous round yellow. So like that, so many things you can find out from this particular checkerboard or Punnett square. So sometimes I found children trying to write the genotype ratio of this one, but to be frank, I'm telling you, for CBSE level till 12, generally genotypic ratio of F2 is never asked. So no need of bursting your head for that one. All right, so that's all about the dihybrid cross, which is done by our grade scientist Gregor Johann Mental, and the presentation goes like this. And if you have given this margin and arranged everything and nicely presented, good, and there you get your 5 marks complete in your pocket. That's all for this particular day. And if you have never subscribed my channel, yes, you can go and subscribe right now, so that you get all the topics without any missing. And near to that, the bell button, please enable, so that you never miss any notification from me. Okay, so till the next video, see you, bye.